Welcome back to the Top T Testosterone Fundamentals course. In this video, we're going to discuss the testosterone production pathways and more specifically just the HPG axis testosterone production pathway. There is another production pathway that we're going to get into that's very similar to this one, but the majority of the details that you need to know are going to be in this video, in this module. So we have the HPG axis, or the Hypothalamic-Pituitary-Gonadal axis, and then we also have the catecholamine pathway, which we're going to talk about in the next module. The majority of your testosterone is produced in this way through the HPG axis, and the other pathway is mostly for quick spikes when you need testosterone in the moment, like in competitions or if you're fighting or things like that. That's when the catecholamine pathway takes effect, but the majority of the testosterone in your body is produced through the HPG axis production pathway that we're going to describe right now. So this page in the ebook, guys, will go through everything in complete detail for you to make it easier for you guys to visualize while I explain this. I'm not going to read this page for you word for word. I'm just going to come to the next one and just explain the chart. So everything starts up here in the hypothalamus. Basically, what the hypothalamus does is it recognizes the amount of estrogen that you have, and by the amount of estrogen you have, there's lots of estrogen receptors on the hypothalamus. It uses that to gauge how much GnRH, or gonadotropin-releasing hormone, to release or produce. So once that happens, there are blood vessels where the GnRH can go specifically from the hypothalamus directly to the anterior pituitary, and in response to this GnRH, the pituitary will release two hormones. One is luteinizing hormone and the other is follicle stimulating hormone. LH, or luteinizing hormone, is the one that will signal to your balls to produce testosterone, and FSH, or follicle stimulating hormone, is the one that is going to tell your balls to produce sperm. Luteinizing hormone goes to the Leydig cells, which produce testosterone, and follicle stimulating hormone goes to the Sertoli cells, which produce sperm. So once the anterior pituitary releases these hormones, they go into blood circulation and eventually get into your testicles, where that communication takes place. Now, once the Leydig cells in your testicles are stimulated by LH, they begin the process of converting cholesterol into androgens. So cholesterol is the precursor to all androgen and estrogen hormones. So let's take a look at this here. These are just some of the conversions that take place. Cholesterol will get turned into pregnenolone. Pregnenolone will then be turned into DHEA and progesterone. Progesterone will also go to the adrenal glands and be used to create cortisol, but the DHEA and the progesterone will both be converted as well into androstenedione. Androstenedione can be aromatase into estrone, which is an estrogen, but also converted into testosterone. And that is how we create testosterone. Now, once we have testosterone circulating around in our blood, we have our body fat, aromatase, converting testosterone into estradiol, which is another estrogen. And then we also have the 5-alpha reductase enzyme converting it to 5-alpha dihydrotestosterone, otherwise known as DHT. DHT is estimated to be 3 to 6 times more powerful than testosterone. Now, on your blood work, if you guys get your blood work done and you look at the levels, you'll see these three amounts, most likely, especially if you're in the US. You will have total testosterone, bioavailable testosterone, and free testosterone. So total testosterone is the total amount of testosterone that is in your blood at the time that they took your blood. Now, some of that is bound to SHBG, some of it is bound to albumin, which means it's not very bioavailable. Now, bioavailable T refers to the amount that is bound to albumin and not SHBG, because SHBG is a much stronger bond than albumin. So bioavailable T is like weakly bound testosterone as opposed to strongly bound testosterone by SHBG. And free testosterone refers to the amount that is completely unbound. Usually 1 to 3% of your total testosterone is completely unbound by everything and is free to bind to androgen receptors. Think of the androgen receptor like the outlet, and the testosterone, or the hormone in general, like the plug. So the androgens need to plug into the androgen receptor. DHT and testosterone both plug into the same receptors, and in fact, cortisol is also an androgen and also plugs into those same androgen receptors. As well as some of these precursors, androstenedione, DHEA, progesterone, all of these are their own hormones that all also bind to androgen receptors as well. So I'll go up here and give you guys some of the specifics. So now that you guys have seen that graph and you kind of understand it, I'm going to read through it just for the guys who want the audiobook version to hear my voice read this. So here we go. It all begins in the hypothalamus. Gonadotropin-releasing hormone, or GnRH, a hypothalamic neuropeptide, is a main regulator of male reproductive function in mammals. GnRH is required for reproduction. GnRH is secreted from the hypothalamus and binds to receptors in the anterior pituitary. GnRH regulates the production and release of luteinizing hormone, or LH, and follicle follicle-stimulating hormone, FSH. Gonadotropins, controlling spermatogenesis or sperm production, and steroidogenesis or the creation of steroid hormones. Luteinizing hormone or LH is a hormone that is co-created along with follicle-stimulating hormone by the gonadotropin cells in the anterior pituitary. Luteinizing hormone is a part of a neurological pathway comprised of the hypothalamus, the pituitary gland, and the gonads. In this pathway, or the HPG axis, LH release is stimulated by gonadotropin-releasing hormone or GnRH. LH causes the Leydig cells of the testes to produce testosterone. Follicle-stimulating hormone or FSH is a hormone produced by the anterior pituitary in response to gonadotropin-releasing hormone or GnRH from the hypothalamus. FSH is a glycoprotein dimer with alpha and beta subunits. The beta subunit is unique to FSH, while the alpha subunit is the same as in TSH or thyroid stimulating hormone. FSH causes the Sertoli cells of the testes to produce sperm. Now, LH and FSH are made from similar genes and thus have similar properties. They're both glycoproteins made up of an alpha and beta subunit. The alpha subunit is the same between the two hormones, and the beta subunit of each is different and gives each hormone its biological specificity. Specifically, the alpha subunit of LH is made up of 92 amino acids, and the beta subunit is made up of 120 amino acids. GnRH release occurs in a pulsatile manner with low pulse frequencies stimulating more FSH production and high pulse frequencies stimulating more LH production. FSH receptors in the testes are found in the Sertoli cells, and LH receptors in the testes are found in the Leydig cells. Sertoli cells are the nutritive cells present inside seminiferous tubules. They provide nourishment to the male germ cells. Leydig cells are present outside of the seminiferous tubules in the interstitial spaces. They secrete androgens. The system described up to this point is known as the Hypothalamic-Pituitary-Gonadal axis, or the HPG axis. When LH binds to the Leydig cells in the testicles, it signals for testosterone production. The Leydig cells take cholesterol and convert it to pregnenolone, which is then converted to DHEA and progesterone. Progesterone is the precursor of cortisol and androstenedione. Androstenedione can be converted to testosterone, estrone, and estradiol, highlighting the critical role of progesterone in testosterone synthesis. DHEA also converts to androstenedione, which is converted by the enzyme 17-beta-hydroxysteroid dehydrogenase into testosterone. Testosterone is then converted to either DHT, or 5-alpha dihydrotestosterone, or estradiol. Testosterone is converted to DHT in the testes and in the prostate by the 5-alpha reductase enzyme. Up to 10% of testosterone is normally converted into DHT. DHT is more powerful than testosterone, estimated 3 to 6 times more powerful than testosterone. It attaches to the same sites as testosterone, the same androgen receptors, but more easily than testosterone does. Testosterone is converted to estradiol, and androstenedione is converted into estrone. Both are estrogens by the aromatase enzyme. In men, 44 to 65% of the circulating plasma testosterone, meaning the testosterone in your blood, is bound to sex hormone binding globulin, or SHBG, and 33 to 54% of it to albumin, and approximately 1 to 3% of it is available as free testosterone, named as such because it is free to leave capillaries and bind to receptors. Bioavailable testosterone on your blood work refers to the testosterone being bound to albumin and not SHBG, considering that albumin is a looser bond that is more easily broken compared to that of SHBG, or sex hormone binding globulin. Testosterone has multiple pathways in the body. The slower pathway is through the genomic or androgen receptor pathway, the pathway through which we build muscle. This is the pathway that testosterone produced from LH signaling from this HPG axis pathway usually takes. And this is the pathway that I've been describing to you guys so far on this page. A faster pathway is the non-genomic pathway, which usually works through the catecholamine production pathway, which takes effect within just seconds or minutes. This is the pathway that gives you increased strength, concentration, mood, energy, and feelings of overall well-being. We're going to discuss the differences between the genomic pathway and the non-genomic pathway in a future module in this section of this course. So don't worry, we'll get into that in detail as well. When we look at different tools for achieving top T or top tier testosterone levels, we look to classify them as production enhancers, acute T-spike stimuli, aromatase inhibitors, 5-alpha reductase enhancers, or T-freers. So production enhancers are things that increase production over time. One of these would be sleeping enough. Another one would be like making sure you don't have nutrient deficiencies. These are all production enhancers. These are really things that optimal production relies on, and when these things are deficient, testosterone production goes to shit. Then we have acute spike stimuli, things like training, things like seeing a beautiful beautiful girl having sex, these are acute spike stimuli. We're going to get into acute spikes in more detail in a future module as well in this course, so I'm not going to go into super detail about it right now, but we will in this course. Aromatase inhibitors are things that stop or at least slow the conversion of testosterone into estradiol and androstenedione into estrone. That's another one that we're going to talk about in detail in another module in this course, so I'm not going to go into super detail about it right now. 5-alpha reductase enhancers, we're going to go into that in the DHT module, but those are things that help the 5-alpha reductase enzyme convert testosterone into DHT. And then we have T-freers, which are things that lower sex hormone binding globulin, or SHBG, and free up more of your testosterone that you already have. So that's it for this module, guys. In the next one, we're going to talk about the catecholamine production pathway.